Okay, Bio, back at it again, Chapter 7, Membranes. Okay, so over the last couple days, you guys have been working on a project where you're developing models of the membranes. You're then taking these models and then showing how certain functions take place. So we're going to zip through this real quick. Hopefully this will help you a little bit with some study, and hopefully this will actually generate some questions from class. So obviously, we know that membranes are the predominant structure of membranes are phospholipids. We know that phospholipids have a polar head and a nonpolar tail. They move continuously in the membrane, hence the term fluid. They move from side to side quite often, but they almost never flip from the outside to the inside. And the reason why is because the hydrophobic areas, which are here, and the hydrophilic areas, which are the little silver ball things, they have a hard time crossing into each other's area. So, for example, the hydrophilic phospho head does not want to plunge down into this hydrophilic region down here. Hydrophobic region. So this is very rare to see the flip-flop like this. We talked a little bit about things that increase fluidity. We talked about this a while ago. When you have more unsaturated phospholipids such as these with the bends in them, they tend to be more fluid. When they're close together, they tend to be more viscous. This also, you see this obviously in different types of cells. So some cells are going to have a higher viscosity, or they're going to be less fluid. They tend to allow less things in because there are tight gaps between them. We see this in like the skin, whereas parts of your intestines might have more space between them. Temperature makes a big difference. So obviously, things that are colder don't have as much kinetic energy. If they don't have the kinetic energy, they don't have the ability to move, let's say. So you're going to find that cold obviously is going to be more rigid than warm. Excuse me. And the last piece that affects it is cholesterol. The more cholesterol that's embedded within the membrane, notice that it's in the hydrophobic area down here. So the more cholesterol you have in here, the more rigid a membrane is going to be. Oh, here's an example. You should be able to name all these parts. We have integral proteins. Oops. We have integral proteins here that go all the way through the membrane. We have peripheral proteins found just on one side, like here, or the other. We have our cholesterol, phospholipids. We'll get more into this a little bit, this extracellular matrix over the course of the next week or so. Cytoskeleton, this is on the inside. These proteins work as attachment points for the cytoskeleton, which is pretty cool. You also have markers, and these markers are going to be glucose molecules that are attached in some type of fashion like this. Sometimes they use the word oligosaccharide, so it's some type of sugar. These are going to be glycoproteins that are proteins with the glucoses. So here's another glycoprotein. And then glycolipids will have the glucose along with the phospholipids. So glycolipid, okay, so on and so forth. Once again, those all work as different types of markers or receptors on the outside of cells. So peripheral proteins, once again, they're not embedded within the membrane. They're either on the outside, they're on the inside, or sometimes they're even in the middle where the hydrophobic tails are. Integral proteins, they go all the way through. So here's an example, sometimes called a transmembrane protein. They go across the membranes, and hence the trans, like transatlantic ships across the Atlantic. So here's a really great view of integral proteins. So we talked about how proteins have three shapes: primary structure, sequence of amino acids, coils and sheets. Here we see a bunch of coils here, all right, which gives us a three-dimensional shape. So here's the ribbon or sequence of amino acids. Here are the coils, maybe some sheets down here, if you will. The parts that are embedded within the membrane here, like you see the part that's across the right here where it's hydrophobic, that section, that whole entire section of the protein is actually hydrophobic. So it's got R groups on it, or functional groups attached to it, that make it hydrophobic. And then your hydrophilic parts are on the outside. So you can imagine if you were tiny little molecules outside, you could easily fit through these little tubes that are here that the coils are made of. So this is one of the ways that proteins can work. Integral proteins is they can work as tunnels, if you will. Any change in temperature, salt concentration, pH will affect how these, the shapes of these, and we'll talk more about that in Chapter 8. Protein function. So here are the 6 functions of proteins. put a little asterisk next to the slide. This is one you need to know. So they provide framework for cells. They aid in transport. They work as enzymes. They can be signals or signal transduction, meaning I take a signal from one place and send it to another. They can also aid in intercellular joining and a cell-to-cell recognition. So hey, this is my cell. This is your cell. So here's a diagram of these pieces. So transport, they can either be active transport or passive transport. Active would be from less to more. Passive would be more to less. They can work as enzymes. All right. So an enzyme has a shape that fits certain things. In this case, the triangle fits in that little spot there. And this enzyme or this purple protein here would take this particular substrate in, would do something with it, spit it out, and change its shape. The second enzyme would then take that particular shape and do something with it. So we'll talk about these enzymatic reactions that take place inside of membranes. Signal transduction. So you can have certain types of proteins that are receptors. So they're looking for certain molecules outside of the cell, and then they'll send some active signal in the cell based on the type of molecules you have here. Pain's a good example of this. So there are molecules that our body makes that would tell us that we're feeling pain somewhere. If you go to the dentist and you have a tooth pulled or a tooth drilled, they give you something called Novocaine. And what Novocaine does is it blocks these signals so that you can't feel the pain molecules. Super cool concept. Intercellular joining, proteins can stick to each other to make one cell stick to another. Cell-to-cell recognition. Some of these, this would be a glycoprotein on the outside of a cell, matches with the protein of another cell, so they know where they are. And they also are the points for either intra, so inside the cell, or extracellular connections. So carbohydrates, once again, here's that term oligosaccharide. It happens to be a bunch of carbohydrates, usually glucoses that are highly branched, meaning that they go, if you look at this one here, it's branched in two locations. So instead of being a 1-4 linkage, it's got kind of a 1-4 and then something else going on there. Hopefully remember that the 1, 2, 3, 4, 5, 6 has to deal with the carbons. And they tend to be covalently bonded to the proteins that they're associated with. So this, once again, you can have glycoproteins, glycolipids work as markers. This is super important in blood types. We'll talk about this a little bit in class. There are 4 blood types: A, B, O, and AB. In reality, there's only actually 2. It's A and B. So A, you have A proteins. B, you have B proteins. AB, you have both proteins. O has no proteins, which is why O is a universal donor. And once again, we'll draw some examples of this in class. Okay, it looks like here's the end of this particular PowerPoint. We'll get back to this momentarily.